I remember hearing when I was a kid that there was a successful cloning of a sheep. Do you guys remember that? A few months ago, as I was crafting myself a beautiful homemade beef burger, I realized that I haven't heard anything about cloning since then, and I wondered why. If we cloned a sheep all that time ago, then surely the cloning science is much more advanced now. Why is there not even a debate about the morality of cloning, like we have with AI right now? So I did what everyone in this day and age of busy lifestyles and endless entertainment at our fingertip does. I put looking into cloning on a to-do list and forgot all about it. Until recently. And let me tell you about it. If I type cloning in Google, here's what I get. Cloning is a technique scientists use to make exact genetic copies of living things. Genes, cells, tissues, and even whole animals can all be cloned. There are three types of cloning, and a great segue to the first type is the following bit. Some clones already exist in nature. Single cloned organisms like bacteria make exact copies of themselves each time they reproduce, which is like if a Targaryen didn't need another Targaryen to make a new Targaryen. Not very genetically diverse, but when it comes to cloning, that's pretty much the point. Alright, real quick, because I understand this is not the cool sci-fi cloning that we're all interested in. It's called molecular cloning, and the way it works is we take a part of whatever we'll use for cloning. So bacteria or yeast. We remove a part of it, we insert what we want to clone, like human insulin for example, and we then put that part back in the bacteria or yeast, and as it naturally clones itself, it also clones the human insulin, and we can now sell it to diabetics. So yes, this type of cloning is widespread, and if you take insulin, you're making use of it. Before molecular cloning, we used to take insulin from cows and pigs, which could cause allergic reactions because it's not human insulin, and was also less abundant. Thanks, molecular cloning! But what about actual cloning? What about Star Wars? What about all the other stories with many clones in them that I can totally name right now but choose not to? Does Altered Carbon count? Anyway, that's not possible. At least as far as making up a grown-up clone, it's not possible. What we can do is crappy twins. Maybe. This is how it works. We take an egg cell from a female specimen and we remove the nucleus in it. The nucleus is the bit of the cell that contains nearly all of the cell's DNA, so it's pretty much an empty egg at this point. Then we take the nucleus from a cell of the target we want to clone, which could be from muscle, skin, bone, etc. I believe pretty much any cell that's not a stem cell would work for that. We then insert the nucleus we took from the target that we want to clone, and we put it in the empty egg cell. This process, and I'm speaking layman here, confuses the egg cell and makes it think that it has been fertilized, so it starts replicating and it forms an embryo, which is a collection of cells. Alright, for the second and third types of cloning, this part is the same. The only thing that changes is what we do with the embryo. In therapeutic cloning, we make use of the stem cells of this embryo and we stimulate them to grow any tissue that we want for the purpose of transplant, or whatever. And since the embryo has the DNA of the target, that means their body will not reject the tissue that we make for them. But how do stem cells do that? Well, they are the body's raw material, so to speak. They are cells from which all other cells with specialized functions are produced. So that's what happens. They repurpose themselves into something more specific. And how do we stimulate them to do that? Science? There are no reliable ways to do that based on my research. We're still sciencing that part out. But some things that are being done to try and stimulate the stem cells to do what we want are introducing relevant proteins to the stem cells to nudge them in a certain direction. Or we co-culture them with other cells and we hope they just join the club. Or we create a 3D structure that mimics the relevant tissue environment and we hope the stem cells get the memo. In theory, functional therapeutic cloning seems very potent. Here are some examples of what it can be used for, and the next one is a quote. These cells could be used to treat a variety of conditions, including Alzheimer's disease, Parkinson's disease, diabetes mellitus, that word over there, stroke, and spinal cord injury. In addition, stem cells could be used for in vitro, meaning laboratory, studies of normal and abnormal embryo development, or for testing drugs to see if they are toxic or cause birth defects. That's pretty neat. Of course, there are ethical concerns when it comes to human cloning, as with everything, but the main reason we haven't heard much of anything about cloning, despite how revolutionary it seems, is because to date there is no confirmation that scientists have successfully produced human embryo through cloning. There are some claims, but no proof. But we cloned animals, right? How come we can clone animals, but not humans? I'll read you a long quote from one of the articles that I read about that. From a technical perspective, cloning humans and other primates is more difficult than other mammals. One reason is that two proteins essential to cell division, known as spindle proteins, are located very close to the chromosomes in primate eggs. Consequently, removal of the egg's nucleus to make room for the donor nucleus also removes the spindle proteins interfering with cell division. In other mammals, such as cats, rabbits, and mice, the two spindle proteins are spread throughout the egg, so removal of the egg's nucleus does not result in loss of spindle proteins. In addition, some dyes and the ultraviolet light used to remove the egg's nucleus can damage the primate cell and prevent it from growing. Okay. No science fiction yet, got it. But how does cloning work? Actual cloning? Very simple. You take that embryo that we created as described previously, and you put it inside the person who donated the original cell where we put the nucleus of the person we want to clone. She then has a baby, and that baby is nearly identical to the person who donated the nucleus. Why nearly? Well, because mitochondrial DNA comes from the baby carrier. So unless the person who provided the egg cell and who carried the baby is either the target we want to clone or the biological mother of the target we want to clone, there will be differences. And well, even in that case, there can still be differences because of mutations, developmental variations, and varied imprinting effects can all introduce differences. In case you were wondering, yes, that means that identical twins are better clones of each other than anything we can do in a lab. Regardless, clones are still pretty close to being exactly the same biologically speaking. We've managed to clone sheep, cats, mice, and whatnot, but why? Why do we care about cloning animals? I'm going to quote again. Reproductive cloning may enable researchers to make copies of animals with the potential benefits of the field of medicine and agriculture. For instance, the same Scottish researchers who cloned Dolly, which is the aforementioned sheep, have cloned other sheep that have been genetically modified to produce milk that contains a human protein essential for blood clotting. The hope is that someday this protein can be purified from the milk and given to humans whose blood does not clot properly. Another possible use of cloning animals is for testing new drugs and treatment strategies. The great advantage of using cloned animals for drug testing is that they are all genetically identical, which means their responses to the drug should be uniform rather than variable as seen in animals with different genetic makeups. Second quote. After consulting with many independent scientists and experts in cloning, the US Food and Drug Administration decided on January 2008 that meat and milk from cloned animals such as cattle, pigs, and goats are as safe as those from non-cloned animals. The FDA action means that researchers are now free to use cloning methods to make copies of animals with desirable agricultural traits, such as high milk production or lean meat. However, because cloning is still very expensive, it will likely take many years until food products from cloned animals actually appear in supermarkets. If this is the case, and this time we have actually managed to clone animals, then why aren't we hearing anything about it? Surely there would be a huge discussion about cloned products and cloning animals. Well, let me throw some numbers at you. Dolly, the sheep that was cloned, was the only clone to be born alive out of a total of 277 cloned embryos. Also, as an aside, she lived 6 years, which is half of what is expected of sheep, and she died of respiratory problems. But I believe the shortened lifespan issue was solved as per the following quote. After an 8-year project involving the use of a pioneering cloning technique, Japanese researchers created 25 generations of healthy cloned mice with normal lifespans, demonstrating that clones are not intrinsically shorter lived than naturally born animals. Other sources have noted that the offspring of clones tend to be healthier than the original clones and indistinguishable from animals produced naturally. I won't bore you with the details of telomere length and its effect on aging, but I will say that reading about this experiment got me to check a few more numbers. Natural miscarriage is 10 to 20%, and it happens because the developing embryo or fetus might have chromosomal abnormalities or other problems that prevent healthy development. The body naturally recognizes and eliminates those issues through miscarriage. The cloning miscarriage rate is 50 to 90%, but do take that number with a pinch of salt as I wasn't able to find a good source of that. The miscarriage rate could be caused by a number of reasons, such as reprogramming difficulties. The process of transferring nucleus from an adult cell into an egg cell often leads to incomplete or abnormal reprogramming. This can cause errors in gene expression and development, leading to pregnancy loss. Or underlying genetic issues. The DNA from the adult cell might have accumulated mutations or epigenetic modifications over time. These can contribute to developmental problems and pregnancy loss. Alternatively, cytoplasmic incompatibility. The cytoplasm, excluding the nucleus, of the egg cell might not be fully compatible with the genetic material from the adult cell. This mismatch can disrupt cellular processes and hinder development. In other words, we don't hear much about cloning and it is not widely used because it is very inefficient. Here are a few interesting tidbits I found about cloning. In 2017, 2 macaque monkeys were successfully cloned using nuclear transfer in China. Those are primates, which shows progress when it comes to potential human cloning in the future. Clones do not always look identical. Although clones share the same genetic material, the environment also plays a big role in how an organism turns out. For example, the first cat to be cloned, named CC, short for copycat, of course copycat, what else would it be, is a female calico cat that looks very different from her mother. The explanation for the difference is that the color and pattern of the coat of cats cannot be attributed exclusively to genes. A biological phenomenon involving inactivation of the X chromosome in every cell of the female cat, which has 2 X chromosomes, determines which coat color genes are switched off and which are switched on. The distribution of X inactivation, which seems to occur randomly, determines the appearance of the cat's coat. And lastly, human cloning is banned in most countries due to ethical concerns and high failure rates. There, now you know a bit more about cloning.